Okay, so the three stages of cell signaling, how does this whole process actually happen? It was Earl Sutherland and his colleagues who discovered how the hormone epinephrine acts on cells. And so he's kind of the guy who helped us figure out, oh, okay, these are the three stages. So there is signal reception, signal transduction, and then cell response. And so that is also how this part is going to be broken up. Those like broad categories and then the different details within. But yeah, those are the three overall stages of cell signaling. Some kind of message gets received. That message gets passed along in signal transduction, and then the cell response. Something happens in response. And here's a visual of that. Again, just an overview. So the signal has to get received in signal reception. So whatever signaling molecule that is, whether it's like a neurotransmitter or a hormone, this molecule gets received. Something happens. And then stage 2 is signal transduction. And so this message kind of gets passed along through different relay molecules. It's like relaying this message. And then stage 3 is the cell response. There's actually going to be some kind of response. So in reception, the target cell detects a signaling molecule that binds to a receptor protein on the cell surface. In transduction, the binding of the signaling molecule alters that receptor and then initiates a signal transduction pathway. And this is transducing the message into like different forms. And so you'll see what that means in a little bit. So transduction often occurs in a bunch of steps, a series of steps between all these different relay molecules. And then finally, in the response, that transduced signal triggers a specific response in that target cell. So that's kind of like the overview. And now we'll go into the details. So stage 1, signal reception. So the binding between a signal molecule and its receptor is highly specific. So if you kind of remember with enzymes, enzymes recognize something very specifically, and that's called its substrate. So for receptors, what molecule that it recognizes is called its ligand. So a signal molecule or ligand and its receptor is really specific. So in this little picture at the bottom, this blue protein only recognizes this ligand that fits perfectly there, and then same for the other ones. So that's where the signal gets received, is like that signaling molecule binds to the protein receptor, and it's usually on the cell membrane, but some can be inside the cell. And so a shape change in a receptor is generally the first step in transduction. So the signal gets received, and then as part of transduction, maybe there's some kind of change to the receptor, and that starts to change down the line. So yeah, most signal receptors are on the cell membrane, but there can be some inside the cell. And so that's also how we'll talk about and break up signal reception. So part A, receptors that are on the plasma membrane. So there are 3 main types of membrane receptors, 3 different kinds of like proteins that are there and receive a certain ligand. So there are 3 main types: G protein coupled receptors, or GPCRs for short, thank gosh. Receptor tyrosine kinases, or RTKs for short, and then ion channel receptors. So these I will also break down in the next couple slides. So these are different kinds of receptors that can be in the plasma membrane. So GPCRs, or G protein coupled receptors, are the largest family of cell surface receptors. Most water-soluble signal molecules can bind to the receptor sites on the receptor proteins that transmit information from the extracellular environment to the inside of the cell. So the signal molecules are usually water soluble because, the inside of the cell is pretty soluble and then the outside of the cell. Water soluble signal molecules will be on the outside of the cell and then bind to specific sites on the receptor proteins, and that transmits information from the outside of the cell to the inside of the cell. Without that molecule even coming in, it kind of just shows up, gives its message, and then the cell passes the message along. So GPCRs are cell surface transmembrane receptors. So remember we talked about, where different proteins can be, if they're on the peripheral, like on the inside versus outside, or if it's a transmembrane protein, it means it spans the whole membrane. So these are transmembrane receptors that work with the help of a G protein. So it's kind of like a little buddy that helps. I'll show a picture right now. So G protein coupled receptors. There's a receptor and it's coupled to a G protein. And so G proteins bind the energy-rich molecule called GTP. And if that sounds familiar, it's kind of like a relative of ATP. So ATP is adenosine triphosphate. GTP is guanine triphosphate. So it's, really similar. And they are still pretty energy rich because they have those 3 phosphate groups, similar to ATP. G proteins are all very similar in structure. And these systems, again, they're the biggest family of these receptors, and so they are extremely widespread and diverse in their functions. So here's a picture. So here is a G protein coupled receptor. So this is the receptor, and then this teal oval is the G protein. So the receptor is here, the G protein is close by. It's usually inactive. It has GDP bound, guanosine diphosphate, but it can bind GTP. So when that cell's or this particular receptor's signaling molecule binds its ligand, it's activated, then it activates the G protein by binding GTP. And then now this G protein, the little buddy, can go off and start this whole pathway of passing on this message. So the little buddy, the G protein, will go activate a different enzyme, and then that enzyme might activate something else and start this whole relay race, passing on the message. So the signaling molecule doesn't have to enter the cell. It really just binds at the surface of the membrane and where the protein is, and then enacts this whole cascade of events. So those are GPCRs, the most common family of cell surface receptors. The second class or type of cell surface receptors are receptor tyrosine kinases, or RTKs. So RTKs are membrane receptors that catalyze the transfer of phosphate groups from ATP to another protein. So these receptors, will transfer or catalyze the transfer of phosphate groups from ATP to some other molecule, usually another protein. So a receptor tyrosine kinase can trigger multiple signal transduction pathways at once. And abnormal functioning of RTKs is associated with many types of cancers. So let me show you a pictures. So receptor tyrosine kinases kind of look like this, like there's two parts. And so when the ligands for this receptor bind at the ligand binding sites, they come together. So on their own, they're inactive, but when they bind their ligands, they get activated and then they come together and now they're active. So when they come together and form a dimer, then that's the activated form. And so the activated tyrosine kinase can then catalyze the phosphorylation from ATP to another tyrosine kinase. So transferring the phosphate groups from these 6 molecules of ATP, now this fully activated receptor can go off and do things. So it's a bunch of steps, yeah. Step 1, ligands bind. They join together into the dimer. These regions are active, but not phosphorylated. And then so once they're phosphorylated from ATP, now this RTK is fully activated. And it might go off and activate some relay proteins or inactivate something. Yeah. It depends on like what the message is, what it's going to do. And so that's how RTKs work. They have to get activated by getting phosphorylated. Sorry. And then they can like activate other relay proteins and then enact cell responses. And then, okay, so this should be number 3, sorry. But it's, again, the third of the list of membrane receptors are ligand-gated ion channels. So a ligand-gated ion channel, if you remember that lecture about, active diffusion, diffusion, passive transport, active transport. So a channel lets stuff in and out. And so a ligand-gated ion channel is gated and lets ions in and out. But it has to be opened like a gate. So a ligand-gated ion channel receptor acts as a gate that opens and closes when the receptor changes shape. So when a signal molecule binds to the receptor, then the gate will be opened and allows specific ions through. So for example, sodium ions or calcium ions. So here is a diagram. So here is a ligand-gated ion channel receptor. And so when this ligand binds here, then the channel goes from being closed to open, and now these ions can come in down their concentration gradient. And then maybe the influx of these ions is going to trigger a cell response. And then once the ligand releases and unbinds, then the channel closes again. So ligand-gated ion channel. So I think those, they're all a mouthful, but they are very different from each other. So focus on like what's different from each other to remember them. And so those are different, again, membrane proteins that are on the cell membrane. There are also cells that are inside, receptors that are inside the cell. So intra means inside. So inside the cell. Intracellular receptor proteins are found inside in the cytoplasm or in the nucleus of these target cells. So that means things that can pass through the cell membrane are things that will trigger these intracellular receptors, right? Because how else? So small hydrophobic chemical messengers can cross the cell membrane, pretty easily, and they'll come in and then activate these receptors that are inside the cell. So for example, steroid hormones, steroids are a kind of lipid, and lipids are hydrophobic, and so they can cross the cell membrane because the main, again, remember the main thing blocking some stuff is like whether something is hydrophobic or hydrophilic. So if something is hydrophobic, it can pass through the cell membrane, especially through that middle layer of hydrophobic phospholipid tails. So yeah, steroid hormones can cross the cell membrane and activate these intracellular receptors. And an activated hormone receptor complex can act as a transcription factor, which we'll talk about more like later in the course. But basically, like when this happens, these can turn on or off specific genes from being expressed. So here's a picture. Here is a hormone, in this case aldosterone. And so it is a steroid. So it can come through the cell membrane, activates this receptor protein that's in the cytoplasm, and then now that it forms this complex, it can affect whether genes get expressed through transcription. And then so this is like a short overview of like gene expression. So transcription, and then translation and making a protein. So that was all step one, is the signal gets received somehow, right? So step one is the molecule has to attach to something and receive this message. Stage two is signal transduction. And so cell signaling is usually a multi-step process. It's not just this and then this happens. It's usually like a very multi-step process, which is, as usual, makes things a little bit more complicated for us to learn, but it's for a good reason, it actually happening this way. So why is cell signaling a multi-step process? Because multi-step pathways can greatly amplify a signal. So maybe just a few neurotransmitters will really help this signal that it is going to have a bigger response than you might typically think for these small amount of molecules. Does that make sense? So multi-step pathways can greatly amplify a signal. And it can provide more opportunities for coordination and regulation of the cell response. So the binding of a signal molecule to a receptor triggers the first step in a whole chain of molecular interactions. That activated receptor typically activates another protein. That protein activates another protein, and so on until the protein producing the response is activated. So yeah, it's like a whole relay of this message. And so at each step, when this signal is getting passed on, it's kind of being changed. It's not like a physical one molecule is literally getting passed along. This signal or message is actually being changed into different forms. And so that's being called transduced, like changed. And so that's why it's called signal transduction. It's like it's getting passed on, but it's also getting changed in different ways throughout this whole pathway. So at each step, the signal is transduced into a different form, commonly a shape change in a protein. So when one protein gets activated, there's like a shape change. It goes off and activates a different protein. There's a shape change, and then that activates a different protein. There's a shape change. So each time is kind of getting transduced to a different method in a way. And so typically, phosphorylation and dephosphorylation of proteins are what are used to regulate protein activity. So phosphorylation is addition of a phosphate group to a protein. So just like how ATP can be phosphorylated and dephosphorylated, yeah, those phosphate groups from ATP can be transferred onto something else. And then now that is phosphorylated. So phosphorylation is usually done by enzymes called protein kinases. Remember, anything that ends in ase is typically an enzyme. So protein kinases is kind of saying like, oh, it's in charge of phosphorylating. Anytime you see a kinase, that's what that thing does. It's in charge of phosphorylating something. And then there's a different enzyme that will remove phosphate groups. So dephosphorylation is removal of a phosphate group, and that's typically done by protein phosphatases. You can kind of remember it like phosphatase. It takes away a phosphate group. And then just for reference, here's a phosphate group. So phosphorylation and dephosphorylation are what are commonly used to regulate protein activity. Like if something gets phosphorylated, then it's usually activated, and then that goes off and does something else. And so here is a picture, I believe, on the next slide. Here. Maybe let's come back to this. So yeah, protein kinases, that phosphorylate stuff, they're transferring the phosphates from ATP to a protein. Many relay molecules in a signal transduction pathway are protein kinases, and so this is what's called a phosphorylation cascade. It's like one thing phosphorylating another thing, and then that phosphorylates another thing. It's like a cascade of phosphorylation. And so this is pretty handy. Like this phosphorylation and dephosphorylation system is kind of like a molecular switch. So it turns activities on or off or up and down as required. So here's the picture. So here was step one. The signal was received by this receptor. The receptor is activated. Maybe it activates some other relay molecule, and now this activated relay molecule can activate this protein kinase. And then this protein kinase will take the phosphate group off of an ATP and phosphorylate another protein kinase. Now that one's active and it can keep doing the same thing. And so that's a whole phosphorylation cascade where one phosphorylates the next thing as active, and then that phosphorylates the next thing and that's active, and then eventually there's a cell response. And it's so funny because, so yeah, there's different kinds of pathways even within these like big types. So there's one pathway that if you learn about, the series of protein kinases, like this kinase is called MAP kinase. The next one is called MAP kinase kinase because it phosphorylates the MAP kinase. And then the next one is called MAP kinase kinase kinase. And it's just so silly and it's so funny, and I'm glad that they kept the names like that because it's just funny. It just proves, yeah, kinases phosphorylate something, and so that's why this name would be MAP kinase kinase because it has to phosphorylate this guy. So yeah, that's just an aside. So anyway, that's how signals get transduced into different forms. Like a message is kind of still being passed on, but instead of there just being like this signaling molecule getting passed throughout the cell, like this message is getting transduced into different forms by other things, activating other things, activating other things. And many signaling pathways also involve molecules called second messengers. So these are typically small, non-protein, water-soluble molecules or ions that can spread easily throughout a cell by diffusion. So second messengers participate in pathways initiated by GPCRs and RTKs, so not ion channels. Second messengers will be in these pathways initiated by these kinds of receptors. And again, they're small, non-protein, and water-soluble or ions. So some examples are cyclic AMP. So a molecule called cyclic AMP or calcium ions. These two are very commonly second messengers. So a little bit more, cyclic AMP, we could call it CAMP for short, is a small molecule made from ATP. So if it sounds a little bit familiar, yeah, it is made from ATP. And that's the most widely used second messengers, or one of the most widely used. So here at the bottom, you can see how. So there's ATP, adenosine triphosphate. This enzyme called adenylyl cyclase converts ATP to cyclic AMP in response to an extracellular signal. So this is how it becomes like cyclic. So there's still adenosine. MP is monophosphate. And then it's cyclic because there's 2 carbons. And so it kind of forms like a little ring. Yeah, so it's a little ring like that. It's a cyclic adenosine monophosphate. And so that is a second messenger molecule. And when it's not needed, you can just kind of inactivate it with a different enzyme. Phosphodiesterase will kind of like straighten it back out. And now it's just AMP. So many signal molecules trigger formation of cyclic AMP. Other components of cyclic AMP pathways are G proteins, G protein coupled receptors, and protein kinases. CAMP usually activates protein kinase A, which phosphorylates various other proteins. And then further regulation of cell metabolism is provided by G protein systems that inhibit adenylyl cyclase. So like say you don't want this pathway to keep going, then you'll just inhibit this enzyme from making cyclic AMP so that it can't go off and do different things. So to kind of tie different things together, let's review by looking at this G protein signaling pathway as an example. So in the first part, the first messenger is technically this signaling molecule. So like epinephrine, this is like a first messenger. And then that's why somewhere later it's called a second messenger. So that first messenger, the signaling molecule, the signal's received by this GPCR. The activated GPCR activates the G protein. It's bound by GTP, and so now this G protein is activated. That activated G protein binds to this enzyme, adenylyl cyclase. And then activated adenylyl cyclase converts ATP to cyclic AMP, and so now this is the second messenger. And then the activated cyclic AMP, second messenger, now formed, activates another protein, like protein kinase A, which is going to phosphorylate other stuff, have a whole phosphorylation cascade, and then eventually cell response. So again, it is like a lot of pieces, but just kind of practice like one part at a time and then putting it together. The receptors, these are the different kinds of receptors. These are the different kinds of second messengers, and then the phosphorylation cascade that can happen. So just take it one piece of this puzzle at a time. And so, again, even when you're like, why am I taking this class? I want to do this big scale thing. It all comes together. It's all relevant. So, for example, understanding of how cyclic AMP in the G protein signaling pathways works helps explain how certain microbes cause diseases. So, for example, the cholera bacterium, Vibrio cholerae, makes a toxin that modifies a G protein so that it's stuck in the active form. So then this protein is continually making cyclic AMP, and this whole pathway just like keeps going and is activated. And so this causes intestinal cells to secrete large amounts of salt into the intestines. And then water follows by osmosis. And so an untreated person with cholera can then soon die because of all this loss of water and salt. So knowing how things work in the small term, in the small mechanics, like still makes a difference for like big picture career pathways. And then so another example of a second messenger, again, are calcium ions. So calcium ions are also used widely as a second messenger, actually more so than cyclic AMP. I don't know why this is capitalized. That's my bad. California ions. So calcium ions can function as a second messenger because its concentration in the cytosol is normally much lower than the concentration outside the cell. So there is a concentration gradient, right? If there's a lot more inside the cytosol than outside of the cell, there's a gradient. So a small change in the number of calcium ions inside thus represents a relatively large percentage change in calcium concentration. So here is a figure kind of trying to show that. So the intensity of this green color is basically representing high concentration of calcium. So outside of the cell in this figure, it's like a dark green, showing that there's a high concentration of calcium. And then on the inside of this cell, in the cytosol, it's a lighter green, and so there's a lower amount of calcium ions in here. And then if you look inside a couple of these organelles in this cell, it's dark green, that there's a high concentration inside those organelles specifically, a higher concentration of calcium ions. So there's a higher concentration in the mitochondria, the inner, oh my gosh, why am I blanking? Well, inside the innermost section of the mitochondria, and then inside of the endoplasmic reticulum. So yeah, these kind of concentration gradients are for a reason, and they are in practice on purpose. So a signal that is relayed by a signal transduction pathway can trigger an increase in calcium inside the cytosol. And so pathways that lead to the release of calcium involve these other molecules called inositol triphosphate, or IP3, and diacylglycerol, or DAG, DAG, as additional second messengers. And so these two molecules are produced by cleavage of a certain kind of phospholipid in the cell membrane. So here is a picture kind of showing these. So here's an activated GPCR. The first messenger, again, is the signaling molecule technically. It activates the G protein. The G protein activates something else, phospholipase C. This modifies one of the phospholipids in the membrane, a certain kind of phospholipid in the membrane, modifies it to IP3, which is a second messenger. IP3 will go and activate this IP3 gated calcium channel on the surface of the endoplasmic reticulum. And so once the IP3 activates and opens this IP3 gated calcium channel, now the calcium ions flow out of the endoplasmic reticulum into the cytosol. And now calcium ions are another second messenger. And then they might activate other various proteins and then eventually a cell response.